Hello and welcome to this series of tutorials on the Gliamorph toolkit. We will begin with a quick introductory video on the Gliamorph toolkit and its functionalities. Visualization of cell morphology at high resolution via imaging techniques such as ARIScan microscope have provided a wealth of meaningful qualitative information. However, this has also created a bottleneck in data analysis, where very few tools have been able to convert the qualitative results into quantitative data, which would enable further analytical exploration. The Gliamorph tool is a modular image analysis toolkit. It is designed to perform image pre-processing, selection of regions of interest, segmentation, and cell feature quantification. All of this is performed on three-dimensional images, and each of the functionalities is provided as a separate module, allowing flexibility in its applications for a variety of image analysis subjects and techniques. Throughout this tutorial, we will be implementing the Gliamorph tool on the focus of our research group's work, the zebrafish retina. Zebrafish are an invaluable model for neuroscience due to their rapid development over the course of 24 hours, their similarity to human structures, and the ability to model disease by simulating patient-specific mutations. By creating transgenic reporter lines, we can visualize structures of interest in the fish. Specifically, the retina is a meaningful tissue to study the central nervous system, as it is not only more accessible than the brain, but also formed with a very stereotypical architecture, highly conserved between species. The cells that we will be working with are the Müller glial cells. These play crucial functions in the organization, development, and maintenance of the retina. They are composed of 5 key subdomains, starting from domain 1 and 2, representing the apical regions, to domain 3 being the cell bodies, domain 4 being the cellular protrusions horizontally going through the inner plexiform layer, and ending at domain 5, representing the basal end feet. So for orientation, the area that I'm highlighting here corresponds to the end feet contacting the inner limiting membrane, whereas this region here represents subdomains 1 and 2, hence the apical part of the cell. This elaborate shape and highly branched structure provides points of contact for almost all other cells in the retina, supporting their development and functionalities. Quantification of the Müller glia morphology in the developing, healthy, and malfunctioning retina can elucidate the mechanisms behind the evolution of their function and what happens when these mechanisms break down. Thank you for following this first introductory video. You can now continue with the next few tutorials where we will go through how to implement the microglia tool.